So when we talk about the great plate count anomaly, yes, it is what we see under microscope and what we see after plating out the same sample. Both the things do not give us the result that is reliable, that contradicts each other. Under microscope, we are seeing so many bacteria present, but when it is put onto a media, allowed to grow, what you see is only few colonies growing out of those hundreds which we have seen under a microscope. Right? That is what is the great plate count anomaly is talked about. So most of these microbes which are seen under microscope cannot be grown under laboratory conditions. So why it is that what is seen under microscope is it is not growing on plate. So is it dead? Are some of the cells dead so that they are there but they are not in a condition to grow? That is one possibility. What is second possibility is they are viable but not culturable. VBNCs, the short form which is going to be repeatedly occur in this discussion about unculturable bacteria. So VBNC, they are viable, but they are not culturable. All right. Now, when it is to be culturable, that means the bacteria should find it suitable for its growth. It might have some specific requirements. Some of the bacteria are too fastidious and have demands for special components to be readily supplied to them when they have to grow. That is one possibility, right? So for them, that fastidious nuclear nutrient which they really require may not be available in the media that you are providing for its growth. So you might have to go on changing the media, trying out different combinations, and yet you failed. Then the question remains why and how. We'll talk about that also. The second comes the bacteria is viable but doesn't grow. It fails to grow. So is it in the dormancy state and you need to break that dormancy like spores if they are there, which is a dormant phase of bacteria. We have to give some trigger to that so that they come back into their viable state and vegetative state and start growing and multiplying, etc. So if they are in the dormant phase, we need to adjust the laboratory conditions in a way that the dormancy is broken. The cells are triggered to grow and multiply. All right. So you have to provide a particular nutrient that will trigger this dormancy to vegetative cell growth. Uh, um, I mean, the change of mode. Right, maybe a nutrient, maybe the temperature, maybe the pH. You don't know what the factor is, and that we have to find out while making this dormant cells get into vegetative form. All right. Now another possibility is that the bacteria is present, it is not in the dormant phase, but the total number of cells in a unit volume are very less. So probably if there is one cell of a bacteria of one type and there are 100 cells of another type in a sample that you have collected from the environment, maybe soil or water. When you are putting it onto the plate, the volume that you are taking in your pipette, few microliters, and spreading it out onto the plate, maybe after enrichment also, the one cell will grow to 10 maybe, but hundreds will grow to millions. And when you put it onto the plate, what is the probability of that single cell getting a chance to come into the nutrient plate and grow to give the colony in presence of those which are thousands of other species are growing around it, right? So if the total number, the prevalence of that bacteria is less, again there will be a challenge to bring it into culture and make it visible. So there are different reasons why this can happen and why do we fail to grow some of the bacteria in the laboratory. So there are many genetically distinct phenotypes, and the phenotypic characteristics are really so parallel that they are not uh, um, to be studied to the very much of details, and phenotypically indistinguishable bacteria could be there, right? So maybe the characteristics that we are applying to study that isolate may be limited, and it will be really difficult to say that organism as a different strain or different different species or different genus, right? So Some bacteria are resistant to culture or conventional media. Certain bacteria have fastidious growth requirements. Some are in the dormant phase. Some require specific pH conditions, incubation temperature, or even oxygen conditions. So, as I was giving you the example, Microbacterium leprae, when it is growing inside the body of the host, that time many of the nutrients are provided by the host itself, and you don't know what are the specific requirements which are fulfilled by the host, and you are getting that pathogen growing in the host, but you are not able to grow it in the laboratory conditions. Even escaping mechanisms of the bacteria are there, which could be like just simple thing, like in case of clostridia or even mycobacteria, it is going to get um, escaped from the oxygen levels in the system. So might be there are certain mechanisms which are active when the host is when the pathogen is within the host under in vivo conditions, but under in vitro conditions such inherent mechanism of that bacteria also is not working, right? So even if it is present, even if it is culturable, it won't be growing. So these are the different reasons why you fail to grow these bacteria. Now, what is the proof that bacteria is present? As we talked in the previous discussion, molecular tools are there, right? Which will tell you about the presence of DNA. And if the DNA is there, that means organism is there, the source bacteria is there. So, molecular data will tell you about the genetic information about the bacteria, and that will lead to the conclusion that yes, this particular bacteria is present in the given sample. Of course, you can explore the presence of a particular bacterial species by molecular tools, but are there any limitations again? Yes. For example, uh, uh, in case of uh, proofs that we are talking about, so microscopy is telling you yes, bacteria is present, but is it live or dead? So if it is dead, then again its DNA is there. It will get amplified in the molecular data. You will get the presence of that DNA and the bacteria responsible for it. All right. So even if you are talking about molecular data, we need to confirm whether it is from the alive cell or a dead cell. Right. Many a times uh, it is from the dead cell, and then it will be a questionable thing. Yeah, if the cells are in their metabolically active phase, and even though they could not be replicated on the laboratory media, but molecular data will tell us ki yes, they are present, because the DNA studies are telling us that bacteria is present. But what if the DNA is coming from a dead cell? It's one of the possibilities, rarest possibilities, but yes, is there. Let me share with you one research paper that was presented by Woods et al. In 1987. What they had done was they have attempted to cultivate the unculturable bacteria, and more than 85 uh, uh, bacteria they had found by molecular tools, but they were not growing into the culture media. So have no culture representatives to say. They were there, but they were not on the media. They were seen in the molecular data, but they were not in the culturable form. So, given that these diverse groups must be growing somewhere in the environment, and the environment sample was taken for their DNA to be present, that was checked, sequenced, and the point was driven home that culturing efforts of the last 2 centuries had managed to replicate permissive growth conditions for only a small subset of those total diversity. Right? So, whether the DNA is coming from the live cell or it is coming from a dead cell, that is one case. Even if it is from the mm, live cell, uh, uh, why is it not culturable? Then that remains that we have to talk about it, right? uh, but you have to take into consideration that the molecular tools also have some certain kind of limitations. Of course, sequencing also may not be always what we say 100% uh, 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 correct, There may be certain errors in that also. So all these things are to be taken into consideration when you are talking about molecular tools as methods to characterize or check the presence of uh, uh, unculturable techniques, unculturable bacteria. All right. Now, I think I need to reshare the slide. It is not seen, right? Yes. I hope now it is seen. Fine. So when you talk about uncultivable bacteria, then is it that their growth rate is slow, or they are in the dormancy phase, or their total number in the given sample is less? These things we talked about. Another thing is a concept that is case strategist. What do you mean by case strategist? That is, these bacteria are adapted to limited resources. Now, when they are growing in the natural environments, they are not going to have as much of glucose and peptone and all the yeast extract and all the meticulously all the nutrients that you add to the culture media. They are growing in a very lavish conditions when you are growing trying to grow them in the laboratory media. But do you think in the natural environment, say soil or river water, so much of glucose will be made available to a bacteria around it in its close vicinity so that it can reach to it and utilize it? No, right? So whenever they are growing in their natural environment, they're adapted to limited resources. And when you are trying to grow them in the laboratory conditions, it's a kind of reach shock for them. So much is available that they are not able to sustain in that condition. So limited resources, and because of sources are limited, their growth rates are slow, and that's why they are able to survive. They have a stable existence, but it is not possible to grow them in the laboratory because of these things. So they are referred to as case strategists. And against that, you have R strategists. That is, they are present in a particular number, maybe low initially, but as there is a flush of nutrients, they rapidly grow, multiply, and bloom like anything, and their number is going to be very high. So in a environmental sample that you have collected, you have, say, two different species. One is of K type and one is of R type. When you try to put them in the laboratory conditions, in the laboratory media, what will happen? The K type is a one which is going to be adapted to low nutrient conditions and dividing very slowly. As against R, it is adapted to limited nutrition conditions, but when there is flush of nutrients, it is able to switch over to the another mechanism and is able to bloom is to rapidly multiply. So if this is the case and you are trying to see them in your media, what will happen? Only the R type will grow. K type is there, but its rate of division is so less that it will not be seen in the media. So these two terminologies, that is K strategist and R strategist, I hope is clear to you. So what can we do? So we need to mimic the natural conditions under which they are growing. So you have to modify your isolation media formulations, the substance types, the substrates that you are providing, the nutrients you are providing, their concentrations, their natural resources which would provide that particular nutrient must be chosen. You cannot just go with providing pure glucose directly or maybe yeast extract directly. You have to look for what will be really available to that organism under natural conditions. A similar molecule is to be provided. Similarly, the factors like pH, temperature, and other biotic factors. Now, what these biotic factors does mean? So bacteria, when they are growing in the natural environment, they are not alone, right? They are growing together and are interdependent. Some of them may having parasitic interactions. Some of them may be having some what we say coexistence. that is growing together. Synergism may be there, right? So different interactions might be there. Or another thing is that many a times in a given environment, bacteria are going to be growing there in succession mode. That is, presence of one bacteria is going to provide conditions such that by its own metabolism, the conditions are going to get altered such that some other type of bacteria will thrive on it and will flourish. It will flourish and create the conditions that will help some other strain of bacteria, some other species, some other genera of bacteria to grow. So successively, the flora will keep on increasing in number of a particular species, and it will decline, and another species will grow in number. That is how it goes on. So synergistic reactions or parasitic reactions or succession are the different phenomena which are occurring in the nature as bacteria interdependent as far as their existence and nutrition availability is concerned. Metabolite produced by one may be useful for the second for its existence, for its multiplication, for its growth. Unless the bacteria 1 produces that metabolite, it will not be available to bacteria 2. So bacteria 1 is growing, but bacteria 2 is not getting it. Right? Maybe that factor 1 is produced only after the bacteria 1 grows to certain number and the environmental pH change to acidic. Under acidic condition, that metabolite is produced, then it will be released, then it will be available to bacteria type 2. So it will be start to grow after little late, after certain time. So such conditions are there under natural conditions. How do we know them? Right, we need to explore these intermicrobial what we say interactions, and if we understand those, then we will be able to mimic the natural environment. It is not that just it is seawater media that you have to prepare, so take all the salts in the sea and put it. Okay, fine, it is there. What about the pH? What about the pH it when the seawater is not sterilized seawater, but it is under natural conditions, and all different flora fauna, including higher organisms like man and shark, are also interacting in the same environment? What kind of diverse type of metabolites will be available under that condition, and the bacteria is thriving there? You need to understand that, right? So mimicking is a big challenge. It is not that easy. So biotic factors, when you talk about, you have to take into consideration these microbial interactions and the factors contributed through it. Example could be one of the metabolites like siderophores produced by one bacteria, or pH resulting as as an outcome of growth of one of the type of bacteria that would selectively allow the growth of second one, so on and so forth. All right. Now let's go to the next slide. So this is one another term that we need to learn, that is microbial scout hypothesis. So the different hypotheses, different terminologies we are getting, uh, uh, and we are trying to discuss about that. We are trying to know about those, right? So microbial scout, what does it mean? It was given as early as in just a decade um, back in 2012. Of course, I'm very sure that it was put into words and published in 2012, but it must have been detected and talked quite earlier, because there are many scientists who are working but are not publishing their data so adequately. So coming back to the term microbial scout hypothesis, it suggested that given the same growth medium, a culturable but rare environmental species with limited scout formation might elude isolation for a long time. Therefore, longer incubation is favorable for capturing novel species. It's fine that the factor, the number of the particular species in a given environment. So if the initial number is less, maybe initial number is less is one factor. Its rate of division is very slow. That is another factor. Third is probably it is dependent on the metabolites provided by its bacteria which is coexisting with it. So many factors may lead to its slow growth, and if you are trying to check your plate every 24 hours, 48 hours, 72 hours like that, and coming to a conclusion, no, there is no growth of this particular bacteria, there are chances that you are going to lose that bacteria even if it is present. So is it that you incubate the plates longer? So maybe weeks, 4 weeks, 2 months, like that, you will get more number of organisms. Yes, experiments were done like that. They had inoculated the plates and incubated, and the count was taken on the daily basis for days and months. Up to year time, some people have worked for it. So, of course, the number of colonies, they go on increasing, but are they of different species? Is the mm, species which was not reported in early incubation is appearing after delayed incubation or a late incubation, and you are getting to find that which was not initially present? Is it possible? Is it one of the chance, right? So was it observed? That we need to find out. So long term cultivation experiments is equal to more colony count. True? Yes, true. But long term cultivation experiment equal to isolation of a rare species, a new species, unculturable earlier, that grows. Is it possible? That needs to be talked about. Are we going to get the phylogenetic novelty, or is it going to be the multiples of the same isolates which were seen in the early incubation, only their number grows on and you find them? Or it is like some of them were dormant initially and you were able to knock the dormancy, and it brought back in them into viable phase, multiplication phase, and they appear later, and you get better diversity explored. All these are to be talked about, and that talks about the microbial scout hypothesis. So what is a scout cell? The dormant microbial populations periodically uh, form, they are referred to as scout cells. So dormant microbial populations, they are referred to as scout cells, which are not common phenomena. Right, spore formation is not a common phenomena, right? And every now and then cell is not going to go undergo sporulation, but it depends on starvation and temperature and so many factors together. So scout is newly reactivated cell that has left its dormancy in response to infrequent and essentially random unknown internal events. Unknown, random, but essential events. These are the three words. So it was dormant and it became active. Right? So scout is not a genetic variant. It doesn't differ from the typical growing cell of the population, but it is expression of certain genes. It is expression of certain metabolic pathways that would convert it from dormant to an active cell, inactive cell to active cell. So scout explores the available resources and initiates the transition from dormancy uh, to its active phase. So ये कहाँ से आएगा? How will it get the trigger? Right? If you remember in the studies related to natural gene transfer mechanisms in the bacteria, like transformation or conjugation, we see that there are some natural triggers that are leading to expression of the F plasmid for expression of conjugation and formation of the conjugation breach with the help of F plasmid, for example, or the readiness of the bacteria to take up the DNA from the environment. All these things come as a trigger from the natural conditions, the environment in which the organism is sustaining. So, what kind of such trigger is? How it is produced? Is it due to the rest of the population which is growing around it? It is secreting certain intracellular metabolites. In response to that, the bacteria will show a break of dormancy and will come out of it and start growing as living as a vegetative cell. Right? So it is possible that the surrounding population will secrete some of the metabolites that will act as a trigger or stimulus for these dormant bacteria to come out of its scout phase and start growing. So metabolites are the signals, right? So the signals could be of various types, like autoinducers, AIs, or resustenance promoting factors, which are called as RPFS, short form hai. uh, then it could be siderophores, it could be an enzyme like catalase, right? So these are going to be the metabolic signals which are generated by the bacteria, what surrounds it in the natural environment. And when you are growing it into the laboratory, if those bacteria are present in that plate and are secreting those intracellular nutrients into the media and are available to this scout cell, it will take it up and start growing. All right. So it will not get done so fast and so easy. So longer incubation may be required. Right. So when growth conditions are suitable, then and then only this scout cell will come into its growth phase. Otherwise, for a long time, if it doesn't get these inducer nutrients, so that its metabolic pathways for growth are not getting initiated, then the scout dies and new scout will randomly arise in due course in the same dormant population. That is been observed by people. Various reporters have talked about these scout cells. Is the concept for scout is clear? So this microbial scout hypothesis says that a strain may be perfectly cultivable on the given medium, but it is rare in most of the environments. Its case scouts may elude cultivation for years, making an impression that it is uncultivable. So it is going to be dependent on some of the metabolic signals from its uh, bacteria from its same community, some environment, same niche. Other bacterial species have to help its growth. So the increased efforts may bring such a strain into culture by accident maybe, but without the manipulation of the media formulation. Media cannot do anything. It is the metabolite which is produced by coexisting bacteria would help growth of such scout cells. So you have to identify the bacterial interactions, the metabolite involved in the interaction, and how it triggers the growth of the scout cell. So you have to identify the pairs or the communities which are interdependent and put them in culture together. So if it is that, how are you going to get a pure culture? That is another thing. All right. So we will talk about in details as we further discuss on this what we say concept of unculturable bacteria. But to sum up today's lecture, let me share you the slide that summarizes various approaches that are that have been explored to study the unculturable bacteria. So it includes, for example, natural environment. When you are saying you are mimicking the natural environment, so maybe seawater you are taking as the culture media. From that culture media, you are trying to uh, I mean, from artificial media, you are shifting to partially natural media. That is, the water from the sea only is used as a media component, or soil extract is used, or the soil itself is used after sterilization. So nutrients which are there in the soil will be available like that. But again, it is not going to be 100% success. It is not going to give. But many of the bacteria of alpha proteobacteria, they were isolated from seawater using seawater as a nutrient medium. Then it is a diffusion chamber, that is another technique. Then you have soil substrate membrane systems, SSMS. Then you have iChip, ICHIP. So isolation chips are there, so which are used for isolation of bacteria and have been successfully used to isolate the bacteria belonging to the taxa like Firmicutes and gamma proteobacteria and so on, spirochetes, etc., from the seawater. So magnetic nanoparticle mediated isolation, M- MWI is the method which is used for isolating different bacteria. One of the examples is Burkholdia species. So you have a number of methods that people are trying to explore to make these once unculturable bacteria to be cultivated under laboratory conditions, which right from mimicking environmental natural conditions, then co-culture of bacteria, right? Then diffusion chambers, then SSMS, iChip, or MWI. These are the different what do you say approaches that people have used to attempt cultivation of one's uncultured or uncultivable bacteria. So I think it is clear